Nitrogen. It's everywhere, and we all rely on it. Nitrogen is a crucial element in living cells and can be found in amino acids, the nitrogenous bases of DNA, the energy molecule ATP, and the chlorophyll in plants. But how does the nitrogen that constitutes 78% of our atmosphere make its way into a living ecosystem? Diatomic nitrogen, the gaseous form in our atmosphere, contains a triple bond that renders it extremely stable and inert. The nitrogen needs to be in a much more reactive state before plants can use it, but this triple bond is notoriously hard to cleave. For us to break it requires tremendous heat and pressure. But microorganisms in the soil have a much better way of performing this incredible feat. Nitrogen-fixing bacteria convert atmospheric nitrogen into forms that plants can use all the time under standard conditions with the help of an enzyme called nitrogenase. This is the reaction that converts N2 into ammonia, an important component of fertile soil. Nitrogenase catalyzes this reaction by lowering the high activation energy needed to break the triple bond. While the exact mechanism is still debated, the exploration of a previously overlooked byproduct provides new insight into the inner workings of nitrogenase. Researchers found that the production of hydrogen gas might be the key to the enzyme's power. Coupling the energy-releasing production of hydrogen with the energy-absorbing cleavage of nitrogen likely gives nitrogenase the energy it needs to start the reaction.
The nitrogenase enzyme is made of two components. Component 2 supplies the reaction, while component 1 executes it. Component 2 contains an F cluster, which receives electrons from other proteins that bind here. ATP also binds here and hydrolyzes to provide more energy. Component 1 is where the catalytic magic happens. The P cluster ferries electrons from component 2 to the secret weapon of nitrogenase, the M cluster. This is a unique structure of iron, sulfur, carbon, and a single molybdenum atom, with an oxycarbon for stability. Let's watch this section on the M cluster to see the reaction unfold. The 8 electron-proton equivalents seen in the chemical equation are added one at a time. Each step is called an intermediate. The first 4 intermediates result in this structure. The diatomic nitrogen then donates electrons to these 2 hydrogens and binds to the 4th intermediate, producing diatomic hydrogen and releasing energy. This energy is used to cleave the triple bond and attach these 2 hydrogens to the nitrogen. More electrons and protons are added in the remaining 4 intermediates, and we see production of the first, then the second, molecule of ammonia.
Thanks to nitrogenase, plants and all the organisms that consume plants get access to the form of nitrogen they need. The study of how microorganisms use this incredible enzyme to fix nitrogen has vast implications for agricultural improvements, research into catalysts, and our understanding of the greater nitrogen cycle.